An egg machine, by rejection and selection, assembles a fairly uniform product. By similar complicated processes, the kidneys maintain uniformity in blood and tissues. The urinary system of man consists of paired kidneys, each provided with a large artery and a large vein. The ureter carries urine from each kidney to the bladder, which in turn is emptied by way of the urethra. When a kidney is sliced open, we see the region which contains thousands of minute collecting tubes which empty urine into this central cavity. The urine is actually formed in this outer region. The unit which forms urine is microscopic. A small artery leads to a capillary tuft. Surrounding this tuft is the closed end of a long, tortuous tubule. The blood leaving the capillary tuft flows in this vessel to the tubule, where it divides into many capillaries, which finally converge into a vein. There are about 1 million of these minute functional units in each human kidney. In this cup, the glomerulus, urine formation starts with filtration of certain blood constituents. This filtration can be demonstrated with a reservoir, tube, and membrane. Water slowly filters through. By raising the bottle, thereby increasing the pressure, water filters through more rapidly. The blood pressure in the kidney similarly forces fluid through the glomerular membrane. When we add a colored salt to the water, the salt passes through the membrane as shown by the dark filtered fluid. The membrane is permeable to this and similar small molecules. But within this liquid, there are the large molecules of a black dye which do not filter through, just as the glomerular membrane holds back large molecules like the proteins of normal blood. In somewhat the same way, this gravel sifter lets small particles through, but holds back large ones. In living membranes, this selective process is called semi-permeability. By manipulation of extremely small pipettes under a microscope, we secure for chemical analysis a sample of the fluid from the glomerular cup. In this fluid, salts, urea, and sugar are in the same concentration as in the blood plasma, but there are normally no proteins. Glomerular fluid is essentially a protein-free filtrate of the plasma. We shall now take a sample at the end of the tubule. In comparing its composition with that of the glomerular filtrate, we find that all the sugar has been reabsorbed. The great concentration of salts, and especially of urea, points to a very large reabsorption of water. More than 95% of the water filtered by the glomeruli is reabsorbed into the blood by the tubules, especially near the far end. Normally, all the sugar is taken out of the filtrate by the tubular cells and is returned to the blood. Urea and other waste products of metabolism are not reabsorbed. Hence, these are greatly concentrated in the urine. These processes, and in fact many others not shown in this simplified diagram, go on simultaneously in the healthy kidney. When we balance the urine against an equal volume of glomerular filtrate, the urine is heavier. This shows that the filtrate has been concentrated. We will study on this anesthetized animal some factors affecting urine formation. We have inserted a collecting tube into each ureter. We record each drop of urine on this moving paper. This gives us the rate of urine formation. The pressure of the blood in the animal's artery is transmitted through a tube to this mercury manometer, which also records on the moving paper. This record shows normal blood pressure and normal in this animal. We have withdrawn a quart of blood from the animal. This causes a fall in the blood pressure. Note that the rate of urine formation is decreased, for with lowered blood pressure, less fluid is filtered through the glomeruli. In another anesthetized animal, we inject urea, an end product of protein metabolism. The urine flow is increased. The kidneys remove the excess urea, thus helping to keep the blood composition constant. This takes more water from the blood, hence more urine is produced. A living kidney, even though removed from the body, when perfused with blood, continues for some time to secrete urine in a limited way. Urine formation is essentially controlled by chemical and physical factors in the blood, acting on the living kidney cells, a masterpiece of evolutionary achievement. We have anesthetized 2 dogs of equal size and are collecting the urine from each. The rate of flow is the same. Into a vein of the animal on the right, we inject a sugar solution. Observing the flow of the urine out of these tubes, we see that the rate of urine formation in the experimental animal on the right is much greater than in the control animal. After a few hours, the difference in urine volumes emphasizes this fact. We now take 2 samples, one from each beaker, and test them for sugar. The test shows sugar in the experimental dog's urine on the right, but none in that of the control animal. In the latter, all the sugar that filtered through the glomeruli was reabsorbed by the tubules. In the experimental dog, some of the excess blood sugar is washed out of the body. Excess salts in the blood, like table salt, are removed in a similar manner. Also, by forming an alkaline urine, the kidneys are herbivorous and the essential neutrality of the blood. While in carnivorous animals, the acidifying effect of the diet is counterbalanced by the formation of an acid urine. The kidneys also control the amount of water in the body. On normal water intake and on excess water intake, the same amounts of water are lost in 24 hours through the lungs and through the sweat glands. But the excess water is removed by the kidneys as dilute urine, illustrated on the right. Compare the water loss under normal temperature and under high temperature, while keeping the water intake the same in both cases. A man exposed to high temperature loses much more water through the skin by sweating, and less through the kidneys. Here we have 2 young littermate rats. With the experimental rat anesthetized, we shall remove 1 kidney. 2 months later, both rats are equally healthy and vigorous. The normal kidney has a margin of safety in not working to its and in its capacity to regenerate. This stop-motion photography shows the gradual filling of the bladder with urine. The musculature of the bladder and of the internal sphincter are controlled by these nerves. When the bladder is distended by urine, nerve impulses from the bladder pass to the spinal cord and brain, starting nerve impulses which relax the sphincters and cause contraction of the bladder muscles, which forces out the urine. In the newborn, this control occurs almost exclusively through the spinal cord. Voluntary control through the brain develops gradually through experience. The functioning of the kidneys in keeping a balanced composition of blood and tissues is somewhat analogous to the action of the gyroscope. The gyroscope, once set in motion, maintains its dynamic balance against external forces. Continuous kidney action similarly maintains a balanced composition of blood and tissues in face of the reverse influences of the processes of living in health and disease.